Thank you very much for that kind introduction. I'm happy to be here, honored really to help you celebrate the wonderful accomplishments of the department and to be part of this anniversary celebration. It's really a pleasure to be here, and I only have one small correction. The baseball season is alas over. So good. I'm a neurologist at Mass General Hospital in Boston. It's part of the Harvard system, and I see patients with primarily dementia illnesses. Oftentimes they come to us early on with a subjective sense of memory problems and want to know what the future holds. They oftentimes, as time goes on, develop additional problems. And we do our best to understand these issues from a clinical perspective and, of course, to treat patients with this kind of standard therapies. But what I want to do today is ask a sort of different set of questions with you. Ask what we've learned about the disease over the course of the last decade or two, and what sorts of risk factors, and in this sense I mean very, very strongly biological risk factors, we can understand, and then potentially, and this is a little bit of blue sky, who knows if this could ever really be part of the universe, but if there's some way that we can intervene in those risk factors to try to help our patients. In the United States, we're obliged to put up conflict of interest slides in the belief that anyone reads them. The goals of the lecture are to ask this question about what it is that we can learn about the disease process and the risk factors, the biological risk factors of the disease, and whether or not there's some way that we can at least dream of, in an optimistic universe, intervening in those risk factors. And the primary risk factor I'm going to talk about today is the risk factor that is in some ways obvious to us all. That is that dementia, and especially Alzheimer's disease, has a very large component which is inherited, that it tends to run in families. That if one's parents have Alzheimer's disease, chances are higher than average, of course, that you will develop it as you get older. There's now 20 known genes that seem to impact that risk. They all aren't of this same magnitude, of course. There's 3 very rare genes that cause an autosomal dominant early onset Alzheimer's disease, age of onset in the 40s or 50s. The youngest patient I've taken care of with an autosomal dominant gene is a 27-year-old woman. Just an overwhelmingly devastating illness, yet still very, 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 very rare. The more common genes that influence the likelihood of getting Alzheimer's disease are apolipoprotein E, which accounts for the vast majority of the relative risk, and then 19 other genes, each of which account for just a small amount, but might be important from a pathophysiological perspective. I'm going to focus primarily on apolipoprotein E because that is, as I say, the gene that has the biggest impact, and also the gene that is most commonly inherited in the general population. So across the world now, apolipoprotein E has been studied, and although the relative allelic frequencies of the 3 common alleles, 2, 3, and 4, vary a little bit across populations, the general theme that if you inherit an E4 allele, that gives you a very higher increased risk, and if you inherit an E2 allele, it decreases risk, compared to the most common E3 allele, is true the world round. In China, in the United States, in essentially everywhere but Scandinavia and parts of Africa, the E4 allele accounts for about 25% of the population. So that 1 in 4 of us have probably inherited the E4 allele, putting us at a 3-fold increased risk for Alzheimer's disease. The E2 allele, the rare allele which is protective, present in only 1 or 2% of the population, but even so decreases risk by about 50%. So from an epidemiological perspective, these are huge differences. A 3-fold increased risk is as big an epidemiological change as one sees even with things like smoking and lung cancer. A 50% reduction would be an enormous change. Instead of the 20,000 individuals who in Singapore are destined to have dementia, cut that in half. Obviously, huge impact on individuals and on the healthcare system. So what can we understand about what the role of apolipoprotein E is in the disease? And as I say, I'm going to explore towards the end a few thoughts about how we can take advantage of that knowledge to try to at least someday intervene. I've mentioned that the human genetics of Alzheimer's disease have been studied extensively, in large part with my colleague Rudy Tanzi at the Mass General Hospital. We know that there are these three major genes that are autosomal dominant. They're called presenilin 1 and presenilin 2, and also the amyloid precursor protein, a third gene that's important in Alzheimer's disease. The amyloid precursor protein, it was the first gene to be discovered in Alzheimer's disease. The metabolism of the amyloid precursor protein, not surprisingly because of its name, induces the molecule amyloid, which makes up the senile plaques that decorate the brain in Alzheimer's disease. It's actually those senile plaques that Alzheimer himself first saw in 1906 when he looked under the microscope of patients with Alzheimer's disease. And the molecule that makes up those plaques, the amyloid, is in fact generated by the precursor protein, the amyloid precursor protein. Mutations in the amyloid precursor protein lead to early onset autosomal dominant Alzheimer's disease. Triplication of the amyloid precursor protein, either by itself or in the context of Down syndrome, also lead essentially universally to an early onset dementia, age of onset in the 50s and 60s. And so it's very clear that there's something about the amyloid precursor protein and the generation of amyloid which starts the process of the neurodegeneration of Alzheimer's disease. The presenilin protein is an enzyme which cleaves the amyloid precursor protein to create the A-beta molecule. And so both the substrate and the catalytic enzyme are both involved in Alzheimer's disease. The presenilin, age of onset in the 40s usually, and there are hundreds of private mutations, meaning, unfortunately, hundreds of individual families who have presenilin-derived Alzheimer's disease. Across the world, rare, yet for those families, obviously devastating. Apolipoprotein E, which I'll abbreviate as ApoE from here out, because, frankly, apolipoprotein is just too much to say all the time. It comes in the three genetic variants. They're named 2, 3, and 4. You might ask what happened to ApoE1. I honestly don't know. But they're named 2, 3, and 4. 3 is the most common. 4, 25% of the population, as I said, 2, rare. But the inheritance of the ApoE4 gene leads to a 3-fold increased risk with a single copy, a 10-fold increased risk with 2 copies. It is almost deterministic to inherit 2 copies of the ApoE4. Tau, another gene important in dementia, seems to control progression of disease rather than age of onset. And I'll speak for just a few moments about that. And then, as I say, there are about 16 other genes that have been implicated in the relative risk for dementia or the rate of progression, some of which include neuroinflammation genes and really link, I think, with some of the markers that we heard about earlier, including the responses of the brain to systemic illnesses like diabetes and hypertension. And I think there will be a very interesting interplay between the genes and the environment as we explore more those genetic influences. So each of the genes that have been uncovered in the past decade or so seem to have a slightly different role in this cascade of events that plays out over the course of perhaps a decade before patients are diagnosed with a dementia syndrome, and then the 8 to 10 to 12 years after that diagnosis and as the disease goes inevitably towards death. And those different genes play out in different roles. Some of them are systemic, some of them are within the brain. Again, I'll focus on apolipoprotein E, or ApoE, which is made by astrocytes, of course, the supportive glial elements in the brain, rather than in neurons themselves. And yet the ultimate degeneration that occurs in Alzheimer's disease is loss of neurons. One of the first puzzles we have then is how it is that a protein that's made in astrocytes, and variably made if it's E2, E3, or E4, but always in astrocytes, influences the chance that the neuron that's adjacent is going to live or die as the disease progresses. And that's one of the fundamental mysteries, and I think comes back in a broader sense to this entire mind-brain continuum sort of biology that we've been discussing yesterday and today, in which we have to understand the entire system and how it relates to one another, both at the cellular level, at the neural system level, and at the whole organismal level, in order to see how a disease that plays out over so many years can proceed in this way. Now, the human genetics for apolipoprotein E are absolutely unequivocal. I just looked in the computerized database, the PubMed, about a year ago, and there were over 1,000 papers written about ApoE, each of them demonstrating that ApoE4 is a risk factor, some smaller number of them looking at ApoE2 as well as a protective factor, universally the case across all populations throughout the world. And I just want to emphasize again how profoundly this impacts our day-to-day practice of medicine. Now, when we ask the standard family history question in the clinic, ma'am, did anyone in your family have dementia as they got older? The answer to that question is clearly informative about that person's likelihood of developing dementia. And almost all of the information that question provides is mediated by the presence or absence of ApoE4. So it's a profoundly important genetic risk factor. These are data from Alan Rose's original paper in 1993, and then he redrew them in a paper in Archives of Neurology in 1997. And it's a Kaplan-Meier plot in which the likelihood of having dementia is plotted from, the likelihood of not having dementia is plotted from 0, meaning everyone has dementia, to 100, meaning no one has it, versus age. The average person in the general population is a 3.3. As you can see in this graph here, and this comes from a series of families in North Carolina that he studied, about 50% of the population has developed dementia by age 85 or so. And so it's a devastating disease. It's obviously age-related. If a person has inherited one copy of the 3 allele and one copy of the 4 allele, and the nomenclature then becomes 3-4, the curve shifts so that 50% of the population with that inheritance pattern has dementia almost 10 years earlier. A 4-4, maybe 72 for 50% of the population. And as you get to age 80, almost no one who's a 4-4 remains free of dementia. And so you can see a shift between the rightmost graph, which are inheritance of the E2 allele, which is protective, and the leftmost of almost 20 years. So if we could simply find a way to volunteer those E4 genes and turn them into E2 genes, we would delay dementia onset by about 20 years, which of course from a public health perspective would make all the difference in the world. These are data from our own center mapping over 500 subjects for age of onset. It's the same sort of graph, not fully demented, not demented. This is from the population cohort that we follow. And just as in Rose's data from 1993, we see the same patterns. If you're an ApoE2/3, it's protective with a age of onset that really never in our population gets even to the 50% level. If you're a 4-4, again, it's several decades earlier that you get to the 50th percentile. Almost no one escapes if you're a 4-4. I'm calling your attention to this blue line, which are the 3 individuals who were ApoE22. So these are individuals who don't have a risk allele at all, and they only have the protective allele. Now, our expectation had been that none of those people would get Alzheimer's disease, because we could sort of model this out and calculate it, and their average age of onset would be over 100, and we didn't have anybody in this study who was over 100 years old, and so we thought it would be no one at all. But when the statistician pulled together these data for me, I saw instead that there was this funny graph, and in fact, it looked like there was at least some people who were 2-2s who were impaired. And so in retrospect, we pulled those charts out, and as I say, there were only 3 people who were 2-2, so this is a very small number. But we found this person who was marked in the computer as being impaired, and I pulled up their chart, and they were rated as impaired because they had told their interviewer that they were having trouble remembering things. They were having trouble remembering things for at least the last decade. We gave them formal memory tests, and they did perfectly well. We followed this individual since we did this analysis for another 3 years, and they continued to do perfectly well. In fact, their scores are getting a little bit better year after year with a little bit of practice effect. They greet me by name each year. They ask about my wife. They remember where my kids go to college. I see them once a year for an hour. Yet this individual continues to bitterly complain that they don't remember anything. I only wish I remembered as poorly as they do. And so if indeed this individual has Alzheimer's disease, it's the kind of Alzheimer's disease I want to get, the mildest of all possible forms, in which something seems like you're a little bit jet-lagged, perhaps, but nothing more severe than that. Now, seeing this individual made me wonder, does inheritance of the ApoE2 allele not only protect you from getting Alzheimer's disease, but you saw the graphs, it's not 100% protective. That still, even though it's a delayed age of onset and so forth, there are some people who have inherited the 2 allele, especially the 2-3 individuals, who nonetheless do develop Alzheimer's disease. What happens to them? Do they have a milder ride? Do they have an easier path of Alzheimer's disease? Now, I've emphasized how rare the ApoE2 allele is in the general population, and how even more rare it is in the Alzheimer population. And so in order to examine this question, we turned to a national database called the NAC database, NACC, in which we were able to examine almost 2,000 brains of individuals who had died after having had cognitive testing, and we were able to determine which of them had Alzheimer's disease, which of them, in terms of neuropathology, which of them had some mixture of diseases like vascular disease plus some Alzheimer changes, so forth. We narrowed it down to these almost 700 individuals who had simply Alzheimer's disease as their phenotype. And I'll just show you a little bit of data from this. It's a rather large analysis, but I want to just share with you the raw numbers here from this large data set. The number of subjects is listed up top. The neuropathological changes that occur. If you're an E2, you tend to have a higher chance. Almost half those individuals really didn't have any of those plaques that Alzheimer's said you needed to have in order to have a definition of Alzheimer's disease. If you're a 4-4, only 1% fell in that category. So, this is a 40-fold difference in likelihood to have a lot of plaques. Similarly, for the number of tangles, if you're inheriting E2, only 7% have the most severe number of tangles. If you're a 4-4, 55%. And so even at this sort of raw, just cross-sectional look at the data, it seems obvious that if you've inherited the E2 allele, you have less in the way of neuropathology of the disease. And in terms of clinical symptoms, the Clinical Dementia Rating Scale, Sum of Boxes, which is a global physician-noted measure of community skills, It's a scale of 0 to 18, 0 being perfectly normal, 18 being severely, severely, severely impaired, really unable to do anything, including basic functions on your own. If you've inherited an E2 allele, and remember, these are all individuals who passed away with a clinical diagnosis of Alzheimer's disease. The clinical dementia rating score, sum of boxes, 6, compared to 17, which is essentially maximum. The worst, as I say, is 18, and I think just psychologically, most physicians don't score anyone in 18. At least in our center, that's the case. You want to give somebody a point for something, and so 17 is about as bad as it gets. Mini mental status score, I agree with the comments earlier. This is a little bit of a tedious test to give. The best you can do on it is a score of 30, which is perfectly normal. The worst you can do on a score is 0. Again, this huge difference is a score of 20, which really translates into some mild impairments, some problems with memory perhaps, but getting through life, oftentimes able to live with some supports, but live independently, versus a score of 10, really severely impaired. And so it seems from those data that the inheritance of apolipoprotein E2 would be a good thing from the perspective of Alzheimer's disease. And so my colleague John Groudon, who visited here 2 years ago, is fond of saying that the best way to beat Alzheimer's disease is to have selected your parents wisely. Well, there's a lot to be said for selecting one's parents wisely, and it isn't simply in terms of preventing Alzheimer's disease. But it got us to thinking whether or not there might be pharmacological ways or genetic ways of taking advantage of what's really an experiment of nature, of understanding how it is that ApoE4 and ApoE2, which really differ from each other only by 2 amino acids, and whether we can convert one into the other by adding a small molecule or another protein, or whether we could really do something that's crazy, which is to change the gene itself. To explore that latter possibility, we began to explore how gene therapy might work. And in mice, it's really a pretty well-established technique now. One can create transgenic mice that overexpress one gene or another. One can then add by a series of viral vectors ways to manipulate those genes. This is work done by my colleague David Holtzman at Washington University, in which they manipulated the apolipoprotein E gene of the mouse. And it turns out that if you give the mouse E4 or you give the mouse E2, you have this very dramatic difference in the amount of amyloid that's deposited in these transgenic mice where each of these brown dots or these blue dots in this stain represent an amyloid deposit. These amyloid deposits are, again, of course, the senile plaques that Alzheimer himself described in the first case of Alzheimer's disease. And so it's you don't need the quantitation here to see that if you, in a mouse, put human ApoE4 in, it's a much worse syndrome than if you put ApoE2 in. And this, of course, matches exactly the data from the human autopsy samples that I just showed you. And so it seems as if it's the apolipoprotein E gene itself, that expression of E2 versus E4, can experimentally modulate the amount of amyloid that's deposited. And this at least rules out some of the other possibilities of the human genetics, which is that you've inherited an E4, but you also inherited something else that we aren't measuring. Or you inherited an E2, and you also inherited something else that we're not measuring. Here, the only manipulation is the presence of E4 or E2, and it has this profound effect on the amyloid. In this study by Stephen Paul and his colleagues now at Cornell, a similar set of data. Again, if they introduce green fluorescent protein, an innocuous molecule, apolipoprotein E4 or lipoprotein E2, the amount of A beta, the abbreviation for amyloid burden, goes up again almost fourfold. With E4, it goes down by more than twofold if they give E2. And so the experimental data in the mice that we saw really was very exciting, and we thought, could we develop a technology in which we could at least look forward to a way of doing the same sort of experiment in people? One of the problems in thinking about this in terms of people is that people are, by and large, larger than mice. Now, that seems obvious, but the problem is that the way that you introduce these genes into the mice, the way that David and Holtzman and Steve Paul did, was they took a needle and they injected into the mouse cortex. Now, remember, the mouse cortex is about this big, and so they inject right on one half of it, and with a single poke of the needle, they could infuse the virus to nearly half of the entire brain. And so with 2 pokes of the needle, you could get essentially the entire brain transduced with the virus. And this is, of course, experimentally incredibly convenient. And the mouse doesn't complain. And so it really works out well. If you were to think about doing the same experiment in people, however, you'd take your needle and you'd put it into the frontal lobe, and you'd get a couple of millimeters surrounding that, and that little bit would make ApoE. And then you'd take your needle out, you'd move it 2 millimeters, you'd put it back in, you'd take it out, you'd move it 2 millimeters, you'd put it back in, and about sometime next year, you'd be done injecting the brain. And not that the neurosurgeons would object, because the neurosurgeons, at least at our hospital, seem to delight in these long and complicated operations, and so they might be up to doing it for a year or so. But the patient probably would object to having a hole put into their brain every couple of millimeters. And so we needed to have some approach other than direct injection of the virus into the brain in order to try to think about how to deal with this scale problem, how to deal with the fact that the human brain is just so much bigger than the mouse brain. And also, and a point that we heard earlier, most of the cortex of the brain is affected in Alzheimer's disease. So it isn't simply that we could put the needle into the hippocampus, for example, and fix the whole thing. We really have to be able to treat the entire brain. And in order to approach this, we worked with just a brilliant scientist from the University of Iowa, Bev Davidson, who was a national and really worldwide leader in gene therapy programs. And she had come up with, I think, a very, very clever idea, and then agreed to help us try to implement that idea in this context. The idea was that for a protein like apolipoprotein E, and for lysosomal storage diseases and a variety of other molecules that are secreted by cells, that if we could create a little cell factory within the brain and then make those cells create the protein and have them just secrete it, that we could bathe the entire cortex in that way without having to introduce the gene into every single astrocyte throughout the cortex. And I'll show you the results from the experiment we did with this idea, in which we transduced the ependyma, which is the lining of the ventricles of the brain, with the apolipoprotein E gene. Each of those ependymal cells start to make either E2, E3, or E4. It diffuses out throughout the cortex and as I'll show you, influences the amount of amyloid that's made in the mouse brain every bit as well as those experiments in which the entire brain was directly injected with the virus. And yet the idea here is that it means that there's a single injection, either directly into the ventricle, so a single needle pass, or through the lumbar space, doing what's essentially the same thing as an epidural or a spinal tap, in which you thread a small catheter up this spinal canal towards the fourth ventricle and release the virus there. We've been able to demonstrate that you can transduce the ependyma, which then essentially becomes permanent. So a single procedure, not necessarily even neurosurgical, something which a neurologist could in principle do, although neurologists are really never fond of doing procedures, we could do a lumbar puncture, thread the catheter up, release the virus. And it would be one time and change the genome for that person for the rest of their lives. So does this work? Well, the first thing we did was we used a marker protein, green fluorescent protein, which, as its name would suggest, is a green protein that fluoresces. And when we asked whether or not we could successfully transduce the ependymal cells and have them produce a lot of the protein of interest, we have this illustration. This is the lining of the ventricle. It's a high-power photomicrograph, and indeed the green fluorescent protein is expressed. When instead we introduced apolipoprotein E, and then we asked whether apolipoprotein E could be made. It could be made. We could measure it with microdialysis systems. The apolipoprotein E that was made diffused out into the cortex and actually stuck to the amyloid plaques that were already there. We used an animal model in which the plaques were already present because our patients already have amyloid plaques by the time they would come to need the therapy. And so we wanted to know what happens not in a prevention mode, but after the disease is already established. We used a technique that we had developed some years ago called multiphoton microscopy, in which we can put the living mouse under a microscope and peer into its brain and see living neurons, living synapses, living astrocytes, and see these senile plaques that were present. And we could ask, does the presence of ApoE2 or 3 or 4 change the plaques in any way? Does it make them get bigger, get smaller? Does it make them disappear entirely? That was, of course, our fondest wish, that they would disappear entirely if we gave them E2, and that they would get worse if we gave them E4. And indeed, that's what we saw. This graph illustrates the progression of individual plaques that we look at in an individual mouse over 2 months of time. If we give the mouse E4 or we give the mouse E2, E4, the number of plaques almost doubles. E2, the number of plaques actually statistically significantly decreases by about 30% just over the course of 2 months. More than that, when we look at each individual plaque over the course of 5 months, we see that those numbers continue to deviate, that the E4s continue to get worse, the E2s continue to get better. So from a mouse's point of view, 5 months is about a third of its life. So it's a long time for the gene to be continued to be expressed. And indeed, in some additional analyses, we were able to demonstrate that the health of the neurons and the health of the synapses are actually restored in those mice that developed, that had the E2. And so we had several important points, I think. One is that we could technically transduce the ependyma. It could make a secreted protein and have it really go through the entire cortical mantle. And that in the mice, again, we could replicate what we knew from humans, that the disease became pathologically, and if you'll allow me to extrapolate from neurons and synapses to cognitive state, more healthy in individual mice who are given the E2 gene instead of the E4 gene. We used a technique called array tomography to ask where the ApoE was. This is a technique which is similar to electron microscopy, but gives us a little bit more flexibility. And we were able to ask where the ApoE went to, how it was associated with the amyloid molecules that were present, and how those associated with the synapses. And indeed, I'll summarize the data here, that much like we saw in human Alzheimer's disease, in which we could use exactly the same technique, the ApoE and the amyloid beta associated with synapses much more in the ApoE4 cases than in the 3 cases, and in the mice at least, the E2 are even less. So that the synaptotoxic and neurotoxic effects of amyloid were ablated by the presence of ApoE. And indeed, we were able to show that the health of those synapses was much, much stronger in the presence of the E2 allele than the E4 allele. And so at this point, I'd like to pause, and I'd like to ask everyone in the audience, who here would like to be a mouse so that they could be cured by being injected with E2? it turns out that being a mouse it's really good to be a mouse with neurodegenerative disease, because we've really come up with a lot of ways to fix those mice. But is it possible in the future that we might be able to do something like this in patients? And of course, I don't know. Gene therapy is still in Star Wars kind of approaches, but I want to assure you that there is at least some reason for hope and some reason for optimism. One of the things we worried about, again, was scale. We could transduce the entire mouse brain by doing the ependyma. Could we do it to a larger animal? Well, we now have data from 2 monkeys that we've done with the same technique. And it turns out that it works for monkeys. Now, monkey brains are not as big as human brains, of course, and yet it seems to scale appropriately. So we're encouraged by that. And then in this last slide, I just want to show you some new unpublished data in which we've worked with a colleague of mine, Xandra Brakefield at Mass General Hospital, who has been developing viral vectors that you can inject into the bloodstream, and the virus crosses the blood-brain barrier and it infects the brain. Now, we all know about diseases in which this occurs, in encephalitides and so forth, in which there are devastating consequences of a virus infecting the brain. But Xandra has been able to engineer this particular virus so that it retains the ability to cross the blood-brain barrier and infect the brain, but no longer makes any toxic products. In fact, we have engineered it so that the only product it makes is ApoE. And so we can inject it, again, now in a mouse, into, well, I'm pointing to my arm, but the truth is it's the tail vein. So we inject into the tail the virus with ApoE, and we figured the tail is about as far away from the brain as you can get in a mouse. Inject into the tail, come back a week later, and look at the astrocytes in the brain. And we've been working on this technology for some years now, and I'm pleased to report to you that in our last set of experiments, 20% of the astrocytes are transduced with the apolipoprotein E gene. Those astrocytes are the normal brain cells that make ApoE. They continue to make ApoE, but now instead of making their endogenous ApoE, they make the human E2, E3, or E4. And the initial results are that they do exactly the same thing as human E2, E3, and E4 has done in all these other circumstances. That is, that it has a profound impact on the pathophysiology of the disease, at least in a mouse. And we're now struggling with the ideas of how to take this forward. If we have the technology to change your genotype, to choose everything good about your parents, and also allow you to modify this one little amino acid that maybe you inherited that wasn't so good. Is that a good thing? Is that something we should be looking forward to offering to our patients and trying, if these data are correct, potentially reversing the disease process and preventing further degeneration? It's an exciting time to be participating in these sorts of studies and to be thinking in these terms about how we might be able to build a future in which we can not only work with Alzheimer's disease, but with other diseases in which genetic influences are so important. And it's my belief that this idea of gene therapy, that the barriers to gene therapy are really ripe to being broken down, that we have a better understanding of the genetic underpinnings of disease, by no means perfect, but still enough, hopefully, to make a beginning start. And the right technology for gene therapy, which over the years has had mixed successes, but at least in retinal degenerations, in pediatric ophthalmology, and in several other instances, now has already been in patients for more than 5 years with no untoward effects, and really, at least with the pediatric ophthalmology cases, children who would have gone blind otherwise, without doubt, now can still see miraculous outcomes. And I think it is at least part of the possible futures that we can envision, maybe the best of the futures that we can envision, that simple approaches will be able to allow us to intervene both in the very important ways that we heard about earlier in terms of keeping the body healthy, in terms of keeping the brain healthy, but also to give the brain a little bit better chance, to give it the pathophysiological underpinnings of the chemicals that allow it to continue to stay healthy as time goes on. And so I'll just close with a summary. Apolipoprotein E2 is clearly neuroprotective in patients and in mice. That introduction of ApoE2 in mice can mimic the phenotype of patients. That human and animal imaging data allow direct detection of the plaques, and so there's a way to track whether or not these things are changing. And now we really have to struggle with the important questions about how to go forward with these ideas and whether or not it's possible to impact the genetic factors that lead to disease in our patients. And I'll close just by acknowledging the individuals in my laboratory who work so hard on this, especially Eloise Houdry, who has been taking the lead in the ApoE project, our collaborators, including Bev Davidson and Casey McGuire, and Xandra Brakefield, who with the gene therapy, and acknowledge the support we've received from the United States National Institute of Health and from some private philanthropy. So I want to thank you very much for your attention. I don't know, it looks like there might be a minute or two for questions.
Thank you, Professor Hyman. Any question for him? We are moving in a great new world of gene therapy. Professor Hyman, John Wong, that was a great talk, and I'm just wondering, are there factors which would influence APO2E expression, say either in terms of the immune system or other extraneous factors which could regulate expression, epigenetic or otherwise?
It's a great question. The truthful answer, of course, is that these are very early days, and we worry about those issues, but we have enough experience with some marker genes that we think we understand how to drive the promoter element and to construct the transgene in a way that makes it less sensitive to those sorts of regulatory elements. I always worry when I talk about genetics and I say I think we understand something, because without doubt nature has found ways that we don't know yet to impact these things. But it's something we're very aware of and are trying to monitor. There are many barriers and many challenges yet to go before this is a realistic hope to put into patients, I think. But even if what we do is learn how to manipulate the endogenous levels of E4 and, for example, shut it down, that would be a wonderful outcome as well. So it's exciting times.
Yes, Professor Christian?
I'll speak.
Well, thank you very much. That's really exciting developments. Can I just check? I think that you suggested ApoE2 may reduce pathological load, but in your human studies, you have shown that there are patients who have ApoE2, have pathology, and are still resilient. So what do you think is the mechanism of ApoE2 induce cognitive reserve or brain resistance? Brain reserve.
It's a great question. The best data that we have so far is that the consequences of developing amyloid plaques, for example, on the surrounding part of the brain is a profound loss of synapses in the immediate vicinity of the plaque. It goes down to roughly 10% of the normal density of synapses in the immediate vicinity of the plaque in the average Alzheimer patient. In the patient who has the ApoE2 gene, and nonetheless has the bad luck to have some Alzheimer changes, the area around the plaques that has that profound synapse loss on average, much less severely impaired. And so it looks as if ApoE2 has sort of a dual function. One is to reduce the amount of pathology in the first place, and the second is to make the brain more resilient to the damage that pathology causes.
One last question.
there any pharmacological drug? Are there any pharmacological drug firm that try to develop drug that increase the action of E2 and reduce E4?
I wish that the answer to that question was yes. There were some preliminary studies by a variety of drug companies looking for a small molecule that would change the conformation of E4 to be more E3-like. In fact, the proteins have been crystallized. There have been a variety of attempts. None of those seem to have been able to go forward. It must be a very difficult chemistry problem. It would be absolutely wonderful, in my opinion, if we could approach it with small molecules as well. As much as I'm excited about the sort of the science of gene therapy, it is much more realistic to believe that we would be able to treat patients with a small molecule that would change conformation.
So I'd like to thank Professor Hyman again. He has inspired many of our young researchers. During tea break, you can still ask him a question because time is premium and time is never on our side here. So we thank him once again.